Hello everyone and welcome to another recorded lecture of BIO1 Online. Today we will talk about chapter 10, which is on patterns of inheritance. So we're going to talk about genetics today, which is an exciting topic. Because genetics has a lot of terminology involved, I'm going to recommend using flashcards. You can use physical flashcards or using an app like Quizlet is just as fine. But again, lots of terms means lots of memorization. So do what you think will be best for your brain. So genetics is the science of heredity. So suppose two healthy parents with a family history of cystic fibrosis visit a genetic counselor before deciding to have a child. And the genetic counselor looks at the family's history, looks at the pedigrees, and the counselor tells the parents that their child has a 25% chance of inheriting cystic fibrosis. So now the parents have a choice to make. Should they try to have a child? Is it worth the odds? And how do we know all this information? So this chapter will explain how we understand how different traits get inherited and how we can calculate probabilities of that inheritance. Before we talk about the details of genetics, let's review some basic terminology. We said that a gene is a segment of DNA consisting of nucleotides, A's, T's, C's, and G's, and it carries biological information. That biological information is usually instructions for synthesizing a protein. We have about 25,000 different genes, or 25,000 different sets of instructions on how to make different proteins. These genes are inherited from generation to generation. So if you remember, one of the fundamental characteristics of all living things was that in reproduction, DNA is passed on from one generation to the next. So genes are inherited from generation to generation, and genetics is the study of inheritance patterns and variations in organisms. So this chapter is called Patterns of Inheritance. That's another way to say we're studying genetics. We're studying the patterns of inheritance in organisms. How do genes get passed on from mom and dad to their children? So geneticists can track the inheritance of traits. And a trait is a physical, heritable characteristic. Heritable meaning it can be passed on. So for example, a curly hair is a trait. Straight hair is a different trait. I can physically see these traits, right? They're not invisible. So traits can be easily tracked because they're physically apparent. And the physical traits are determined by the specific alleles of that individual's genome. Remember that an allele is a different version of a particular gene. So we all have, and I'm going to make this a little simple, we all have, let's say, a hair gene, right? And there are two possible alleles. You can either have straight or curly. But remember, you got one hair gene from mom and one hair gene from dad. So maybe both are the straight hair allele, maybe both are the curly hair allele, or maybe dad gave you the straight allele and mom gave you the curly allele. We don't know, right? But regardless, everyone has two alleles, right? We have two alleles in diploid cells. Diploid cells have two copies of every gene, meaning two alleles of every gene. These are other traits. So we said natural curly hair versus naturally straight hair, but you can also have a straight hairline or a widow's peak hairline. That's a different example of a trait. You can be left-handed or right-handed. That's another trait. So traits are determined by different alleles of an individual's genome. So if you remember, in diploid cells, two alleles for each gene are located at a particular locus of homologous chromosomes. So a locus is a precise location on a chromosome, and each known gene has a specific locus. So if you recall, right, in diploid cells, we have two copies of every chromosome, which means we have two sets of every allele, or two copies of every gene. So genes exist in different versions called alleles. So suppose we're looking at either round seeds or wrinkled seeds. The trait we're looking at is the seed shape. So a seed could either be round or wrinkled. There could be a round allele right? A version of the gene of the P-shaped gene. Sorry, this could be a version of the seed-shaped gene that encodes for round seeds. This lowercase allele could be a different version. It's still a seed-shaped gene, right? But this one's a different flavor. It's a different version. This one actually encodes for wrinkled seeds. But different alleles for a particular gene occupy the same locus on homologous chromosomes, right? So since it's a seed-shaped gene, it's going to be on the same location, the same locus, on both homologous chromosomes. So on chromosome, suppose the seed-shaped gene is on chromosome 5. On chromosome 5, this is chromosome 5, right over here you're going to have the seed-shaped gene. And then on the other chromosome 5, right over here you're going to have the seed-shaped gene. Whether they're the same allele or not is another story. So please take the time now to recap about homologous chromosomes by watching the video contained in the ebook. And this is a good video to watch because, again, it just talks about how chromosomes duplicate, the difference between when we say 46 chromosomes versus 23 pairs of chromosomes, talks about alleles, and how meiosis mixes up these alleles. So this is a very, very, very important animation to understand. So we said diploid cells have 2 alleles for each gene. And homologous chromosomes, right? Homologous chromosomes have the same genes at the same genetic loci. So we can say this is a homologous pair of chromosomes. This is chromosome 2 from mom, chromosome 2 from dad, so to speak. And then here we have the gene A locus. Here is the gene B locus, right? So at the gene A locus, the alleles are identical because it's capital A here and capital A here. So these alleles might be identical, right? In the case of the gene A locus, both have the capital A allele. But it's also possible that at the gene B locus, they have different alleles. So look at gene B. In mom, she had the big B, and then in dad had little b. So in this case, the alleles are not identical. I also just want to say that I hate the whole distinction between pink being female and blue being male, so please totally disregard that. That's completely stupid. But the book does it, and I'm just going through it for ease. But that is not a convention that you should associate with, pink female, blue male. So that is bogus. But anyway, continuing. The genotype refers to the set of alleles that an individual possesses. So we said the genotype can be 2 of the same alleles, or it can be 2 different alleles. So the genotype just refers to the 2 versions of the gene that an individual possesses. Right? We said we have 2 copies of every gene, or one copy of each gene. But the 2 copies don't necessarily have to be the same. They can be different alleles, right? They could be different versions. So if the 2 alleles of a gene are the same, like an A, for example, so here's a big A in mom and a big A in dad, we say that it is homozygous genotype. The genotype will be homozygous. Because if these 2 chromosomes end up in a zygote, right, if this is the sperm and this is the egg, and these both end up in a zygote, that zygote will have homozygous, the same big A, right? It'll be homozygous for gene A, for big A, right, in that zygote. Remember, the zygote is when sperm and egg first meet. So in that zygote, you're going to have homo, the same, for the gene A locus. They're both big A. So write this, big A, big A. That's how you say it. Big A, big A. If the 2 alleles of a gene are different, suppose like in gene B, you have big B from mom and little b from dad, we now say that the genotype would be heterozygous. And we would say that's big B, little b. So genotypes can be homozygous if both alleles of a gene are the same, or the genotype can be heterozygous if the 2 alleles of a gene are different. So please make sure that you understand Homozygous genotype versus heterozygous genotype. Right, so if we have two alleles for each gene, a homozygous genotype. If you have a homozygous genotype, that means mom and dad gave you the same allele, the same version of the same gene. So let's say in the case of eye color, let's suppose there are only two alleles, brown or blue. If you got two copies of brown eyes or two copies of blue eyes, that means you're homozygous for eye color. In a heterozygous genotype, we have mom and dad giving you different versions of the same gene. So suppose mom gave you brown eyes and dad gave you blue eyes, that means you are heterozygous for this trait. So take a look here at this set of chromosomes. This is showing a pair of homologous chromosomes that have alleles for 4 genes at the same loci. There's the G gene, R gene, S gene, and T gene. And they're showing you both the maternal and paternal contribution of alleles at each locus. So is the individual homozygous or heterozygous for each gene, G, R, S, and T? So looking over, you don't have to look at this. This is after replication. But in here, what does the genotype look like for each of these? Right? They're all heterozygous, right? They're all heterozygous. There's no two alleles that match up. Now I have a question. Is it possible for a gamete made by this individual to have the alleles lowercase g, r, s, and t? So how can that happen? So take a moment to pause here and tell me how or if it's possible to ever happen. And if you recall, right, during meiosis in prophase 1, there's something called crossing over. So we know what happens in, this is, let's say, meiosis 1. The chromosomes just replicated, and now in prophase 1, there's crossing over. So what happens is homologous chromosomes line up and they physically exchange pieces of genetic information. So suppose there's a crossover right here. This part physically overlaps with this part. What you're then going to have is, I'm going to draw this in blue now. After the crossover event, you're going to have this chromosome that's going to have G, little r, little s, and then this piece of little t. So if this ends up, and then again, that's the crossover, and then of course this one will have, just to keep it, right, that will cross over here, and then take that t. So in theory, it's very possible for a gamete to have little g, little r, little s, and little t, right? And of course, there could be a crossover here, or here too. So there's always possibilities for mixing and matching of different alleles, and that happens during meiosis. So if chromosome 1 from mom has the genes for blue eyes and brown hair, chromosome 1 from dad has the genes for green eyes and black hair, the two chromosomes are what? Pause here. The answer is B. They're homologous. So before we move on, I want to just quickly re-emphasize an analogy that was mentioned in the book that I think can be very helpful. So suppose that when you're born, both your mom and your dad give you 23 cookbooks that contain every single recipe of every food known to mankind, everything you need to live. So these are recipes, these are cookbooks. And for every book that mom gives you, right, there's an analogous book that dad gives you, has the same recipes in the same table of contents. So if you were to open up recipe book 7 of mom and look at page 222, and then you open up dad's cookbook 7 and look at the same page, you're going to see the same recipe for chocolate cake. But maybe mom's recipe has chocolate cake with chocolate frosting, and dad's has vanilla frosting on it. There's still the chocolate cake recipe, but there are minor differences. So in your library, you should have 46 cookbooks total, but they should be paired up into twos because mom gave you 23 cookbooks, dad gave you 23 cookbooks, and cookbook 1 from mom and cookbook 1 from dad both have the same exact recipes in it. But each recipe has variations. Some substitutions, some omissions, some additions, right? But overall, you have access to 2 recipes. 2 different recipes possibly, or 2 same recipes. It's possible that both recipes are identical, right, in both cookbooks. So I hope that makes sense a little bit when we're talking about homologous chromosomes and the genome and how we have 2 versions of every gene. So we said the genotype refers to the alleles of an individual. And those 2 alleles can be homozygous, meaning they're the same, or they could be heterozygous, right? The 2 alleles are different. And now we can introduce the concept of phenotype. So an organism's genotype is distinct from its phenotype, because phenotype refers to the observable characteristics of an individual. So your phenotype, for example, is your hair color, your eye color, the finger length. These are observable characteristics. Some of these are not observable by the naked eye, so phenotype also includes your blood type, which I can't see just by looking at you, but these are observable characteristics. And of course, genotype encodes for the phenotype. So genetic instructions contained in the chromosomes of the genome specify instructions on how to make different proteins, and those proteins make up the observable characteristics of an individual. So this is explained by the central dogma of biology, right? Instructions in the genotype help specify what the observable characteristics of the individual will be. There are some exceptions, right? It's not just simply whatever the genotype says, that's what the phenotype will be, because phenotype is heavily influenced by the environment and other epigenetic factors. And when I say environment, I'm not just saying it's going to be affected by a cloudy day or a rain shower. Right? Environment means what are the pollutants in the atmosphere? What is the level of UV radiation? Is there a high level of stress in the atmosphere? Right? What is the, again, you could go on and on and on. What's in the water supply? Right? What is the financial health of the country? These things affect us in ways we really don't understand yet. So phenotype, observable characteristics, can be shaped by genotype largely, right? But also by other factors, including the interaction with the environment. So in inheritance, we're going to see that the inheritance of alleles, which is part of genotype, determines in part the physical traits, phenotype. Right? And of course, I'm simplifying here. It's not just genes equal phenotype. There has to be more to it. I'm trying to simplify here that genotype leads to phenotype. When you're taking an advanced genetics class, you'll see that there are so many exceptions to these rules. But for now, this is the starting point. So based on what you inherit, based on the alleles you inherited, that's your genotype. And genotype encodes for phenotype. So suppose you have, let's say the big B allele encodes for brown eyes. The little b allele encodes for blue eyes. So if mom and dad both give you a big B allele for brown eyes, the phenotype will be brown eyes. So homozygous brown genotype leads to a brown-eyed phenotype. Similarly, if mom and dad each give you a little b allele for blue eyes, then the phenotype will be blue eyes. If mom and dad each give you different alleles, one gives you big B, the other gives you little b, what would the phenotype be? So take a second and think about what would the phenotype be in a heterozygous individual. And to be honest, I haven't given you enough information yet to make this guess. But the answer is brown eyes. So clearly, just because you have instructions for a gene, right? This person has instructions on how to make blue eyes. But something about the brown eyes overshadowed that blue eye. So still the individual phenotype has brown eyes. It's not one blue eye and one brown eye. So this leads so things like this, right, raise a lot of questions. And there were 2 very misleading theories of inheritance up until pretty recently. Now we understand a lot more, of course. But there is this theory of the homunculus, where if you were to zoom into a sperm on a microscope, under a microscope, some scientists swore that they saw a fully formed miniature fetus in the sperm's head, and that this was the contribution of just the male, and that once the homunculus sperm, the sperm that had a mini human in it, went into the female, it would just basically develop fully. Again, this sounds ridiculous now, but this was a very popular theory. The father contributed just a mini human, and that mini human grew into a full human inside the female. There's another theory called blending inheritance, in which the parental traits become mixed. So if you have a very, let's say if you have let's get an example, a very tall father and a very short mother, the children will be directly medium-sized, right in between. So the blending of traits, right? If your dad had black hair and your mom had white hair, then it would be gray hair, so to speak, even though that doesn't exist. So blending was a common theory, and the homunculus was another common theory. And it wasn't until Gregor Mendel's experiments. Gregor Mendel's an important name to remember. He disproved the idea of blending in the 60s, and it wasn't until then that we really began to understand how inheritance works. Right? We weren't able to solve the puzzles of inheritance until Gregor Mendel's research was published in 1860s. So let's introduce Gregor Mendel, the father of genetics. He knew nothing about DNA, genes, chromosomes, or meiosis, but he was a very good mathematician, and he had a lot of time to do experiments. So he was an Austrian monk in the 1800s, and he studied inheritance patterns in pea plants. And peas are a good choice because they grow very quickly, they develop quickly, they make many offspring, and they also have many traits that appear in two different forms. We call these discrete traits. They're either or, discrete traits. They can be, so he chose traits that were easy to observe and objective. So basically, is the pea yellow or green? It's one or the other. Is the pea round or wrinkled? One or the other. Is it oval or spherical? One or the other. Those are discrete traits that are very easy to observe, right? If you show me a green pea, I'm not going to tell you it's yellow, unless I'm colorblind. So he chose peas because they have a lot of different observable traits, and again, they're easy to grow, cheap, etc. And Mendel's paper, Experiments in Plant Hybridization, was published in 1866 and has become the cornerstone of modern genetics. Basically, what Mendel did was he inferred laws of genetics that allowed predictions about which traits would appear, disappear, and then reappear among different generations. So he used math and statistics to predict which traits would appear and disappear and reappear as we trace many different generations. And he proposed that inheritance involves the reshuffling of particular units from generation to generation. He had no idea that these particular units were genes, but he was the one who came up with this idea that there are these things that get mixed and matched from generation to generation. And of course, DNA, we didn't really understand what DNA was until much later, until 100 years later. Right. Today, we call these particular units genes. So Mendel's paper, here's the microscope used by Mendel, was the cornerstone of modern genetics. Let's discuss Gregor Mendel and his pea plant experiments. Gregor Mendel mated true breeding pea plants in his experiments. True breeding is also known as pure-bred. And what true breeding means is they're plants that produce identical progeny to themselves. They breed true. That means plants derived from green seeds always produced green seeds when they're fertilized with each other. Right? Yellow seeds, the yellow parents only give rise to yellow progeny. Right? These are true breeding. You never see something different. So this is important. True breeding means that they have the same two of the same allele. They're homozygous for a given trait. Right? There's no difference in them. They're homozygous. right? And it's important to know that plants can be fertilized themselves, so self-fertilization, or they can be cross-fertilized. So I can go on and on about this, but very briefly, a pea flower, right, here's peas, here's a pea flower. The fruit is what we actually know as peas. So the fruit comes from the flower. And the flower has male parts, the anthers, and it has female parts, the ovules. So it's possible for this male, the pollen from the anther, to fertilize an ovule, and that would give rise to peas, right, that are self-fertilized. It's also possible for peas to be cross-fertilized. So what I can do is I can take a white flower and remove its male parts. I'm removing the anthers and only leaving the female part. And I'm taking the male pollen from a different plant, a different flower, and putting that pollen on this female part over here. So this is cross-fertilization. We're using two separate flowers. This is like sexual reproduction. And then, of course, you can wait until the flower produces fruit, which is seeds, right? Contains the seeds of the peas. And then from there, you take these peas and you plant them again. And then what grows is the next generation. I would encourage you to listen to this again, because I'll admit, when I first learned this, I did not understand how the generations worked. A cross works, we take the male parts of one plant, put on the female of another. Then that parent will form peas as a fruit. You then take those seeds and you plant them and wait until the next plant grows. Those are the children of these plants. I'd like you to watch this video on YouTube that actually shows somebody cross-fertilizing pea plants. So Mendel made an observation, and he said that not all pea plants are true breeding. So remember that true breeding plants produce offspring identical to themselves. So when he took a green pea and he planted it, and then he self-fertilized a flower from this plant, all of the offspring were always green. When he took a yellow seed, most of the time he would see that this yellow pea plant produced plants with only yellow seeds. However, sometimes when he planted a yellow seed, he got a hybrid plant. So this is a hybrid plant. It outwardly resembles a true breeding plant, but it produces mixed offspring. So there can be either true breeding plants, in which self-fertilization yields offspring with the same seed color as the parent plant, or you can have a hybrid plant, in which self-fertilization yields a mix of different seed colors. And it's important to note that Mendel only saw this hybrid result when he used yellow peas. Anytime he planted a green pea, he always saw true breeding plants that produced green peas. But when he planted a yellow pea, sometimes he saw that all the offspring were identical. They were all yellow. But other times when he planted the yellow pea, he would see the hybrid result. So this led him to do some follow-up experiments. So now we'll discuss Mendel's experiments. And I'm going to start going through this rather fast because I'm expecting you to have to pause frequently. I don't want this to take hours and hours of your time. So Mendel started by crossing true breeding pea plants that differed in only one trait. So he took yellow peas, or specifically he took the pollen from flowers of a yellow pea plant, and he fertilized that. He put that on the eggs of a green pea plant. So this is the cross, right? This is the male symbol. This is the female symbol. So he took the pollen from a yellow pea plant, and he cross-fertilized the eggs of a green pea plant. Right? So purebred yellow peas, and the X means it's crossed with purebred green peas. We call this the P generation because these are the parents. The parental generation is called the P generation. So these are the starting individuals in an experimental cross. So the P generation is the parental generation. Once these parents were crossed, we can examine the phenotypes of the offspring. The offspring of the P generation is called the F1 generation. The first filial generation is the offspring of the P generation. So right now, pause and hypothesize the phenotypes of the F1 Ps. So the F1 generation was all yellow. Is this what you expected or not? Right? When the F1 Ps were planted, they were grown, and then those flowers were self-fertilized to produce the F2 generation. The second filial generation is the offspring of the F1 generation. So now we're taking these F1 plants, these Ps, and we're crossing them to each other. Right? This is called a monohybrid cross, and I'll explain that why in a bit, because it's a cross where there's a single trait, we're only looking at P color, that's mono, with hybrid organisms. These are hybrid organisms, as we'll soon see. So when we take hybrid organisms that only looking at one trait, that's a monohybrid cross. We're looking at only one trait, that's P color, and we're looking at hybrid organisms. These are not pure breeding. How do we know? Because what happens when you cross the F1s, right? The F2 generation gives us a mix, right? In fact, they got 6,000 yellow to 2,000 green Ps, around a 3-to-1 ratio of yellow to green. So again, this is a monohybrid cross because these were hybrid, right? Look at them. They gave mixed offspring, and they were only looking at one trait. So a monohybrid cross looks at a single trait with hybrid organisms. And a monohybrid cross will always give you this 3-to-1 ratio. Ratios are very important in this chapter. You're going to have to memorize some of them, but they'll become more obvious as we progress. So this experiment revealed that the F1 progeny have only one of the parental traits. And he also saw that both parental traits appear in the F2 progeny, but in a specific ratio, a 3-to-1 ratio. Some traits we can say are dominant or recessive. And a dominant, so Mendel referred to traits as dominant or recessive, but we now say that alleles are dominant or recessive. But what Mendel observed was that some traits were dominant to others. Some traits were able to mask the expression of other traits. So in this case, the trait that appears in the F1 was dominant to the other. So in this case, yellow is dominant to green in the case of seed color. Because the yellow allele, the yellow version of the gene, was more powerful or masked the expression of the green P allele color. Right? The green allele disappeared in the F1 generation, and we say that is recessive. I'm going to explain what this physically means again in a bit, right? Because dominant really just means a protein that can exert its effect whenever it's present. A recessive allele encodes a protein whose effect is masked if a dominant allele is also present. So what this dominant allele is doing, it's masking the effect of whatever the recessive allele is saying, right? The dominant allele talks so loudly, so to speak, that it covers up whatever the recessive allele is whispering. So Mendel did this cross in reciprocal. So he used pollen from yellow peas to fertilize eggs from green peas like I showed, but he also did the opposite. He took green pea pollen and he fertilized eggs from yellow peas. And the results from the reciprocal crosses produced the similar results. So this completely disproved the blending hypothesis. Because if blending were true, you would expect the peas to get lighter and lighter green until they're all like a kind of greenish yellow. But this was definitely not true. This confirmed the presence of discrete units of inheritance, and that in fact these units separate from each other. They segregate from each other and they can come back together. during fertilization. So these were very, very impactful experiments. And he didn't stop at just looking at seed color. He looked at seed shape, flower color, stem length, flower position, pod color, and pod shape. So these were seven discrete traits where there's like a one or another. They're antagonistic pairs, either yellow or green, round or wrinkled, purple or white. So in these 7 traits, he saw very similar results. He saw that for each trait, the dominant allele masked the expression of the recessive allele. And we denote alleles by letters. The dominant allele we denote with a capital. The recessive allele is always lowercase. And the first letter designating a trait is often chosen to identify the alleles. So, for example, let's talk about whether a plant is tall or short. So we can specify the allele with T, right? T is plant height, where big T is tall and little t is short. These are 2 different alleles of the same gene. Suppose big T encodes a fully functional allele that can properly cause the plant to be tall. This little t is a recessive allele and it's non-functional. So it's not going to have the power to tell the plant to grow tall. It's not necessarily going to tell the plant to grow short, it's just not going to tell the plant to grow tall. So this is like non-consequential, right? Having a little t is not telling the plant to do anything. Only if you have a big T will it tell the plant to grow tall. So here we have a tall parent and a short parent. Tell me the genotypes and phenotypes of each. And I just basically just told you half of them, right? So the phenotype of this parent is tall, and the genotype is big T, big T. The phenotype of this parent is short, and the genotype is little t, little t. Right? So we have big T crossed with a little t. Tall times short, or crossed with a short. So now I want to try to break it down what we're actually looking at, what this actually means. So remember that each parent is diploid. They have 2 copies of every gene. Right? But we know that when sperm and egg are made, it's only going to have one or the other allele, right? Sperm and egg are haploid. So they only have one version of an allele, either one or the other. They don't have both. In this case, both parents were pure breeding. So they only have one type of allele to pass on to the offspring during fertilization. This parent is big T. So therefore, the gamete has to be big T. Right? This is little t. The gamete it gives has to be little t. Therefore, what will the F1 genotypes be? If this can only give a big T, this can only give a little t, then what will the zygote have? Well, it would have to be big T, little t. Because all the F1 offspring will inherit one big T and one little t. So what is the F1 genotype? We say that F1 is big T, little t. They're heterozygous. And all of them, 100% of the F1 were tall. So which trait was dominant? Tall must have been dominant because there were no short plants in the first generation. So now things get a little more interesting. So this is where we are so far. This is now a hybrid plant. It's big T, little t. So this is a hybrid plant, and we're only looking at one trait. So this is what is called a monohybrid cross. And a monohybrid cross tells us what are the possible genotypes and phenotypes of the F2 generation. Right, so I want to predict if I have this hybrid F1 plant that looks tall, and I self-fertilize it with each other. What are the possibilities? What am I going to possibly get? So now we could just use basic probability and think about it. Well, this is a plant that has big T and little t. Its eggs can either give a big T or a little t. Right? This one has 2 options. It could be big T or a little t. This one over here only had one option. It can only give a big T. But this one could give a big T or a little t. Same thing, right? That other, you're self-fertilizing it. So it could be a big T or a little t sperm. Right? The egg could be big T or little t. The sperm could be big T or little t. So a monohybrid cross is the self-fertilization of this. And after meiosis, the gametes of the F1 plants, right? The eggs must have either a big T or the little t. The sperm will have a big T or a little t. And we know that during fertilization, it's random which sperm and which egg fertilize each other. So to determine the possible F2 genotypes, we make what's called a Punnett square. This is a Punnett square. And a Punnett square is a table that lists all the possible genotypes resulting from a mating. So basically a Punnett square is saying, okay, so here are the two possibilities of the eggs. An egg can have big T or little t. A sperm can have a big T or a little t. But we know that this big T egg can go with this big T sperm or this little t sperm. Right? This big T sperm could go with this big T egg or this little t egg, and vice versa. So a Punnett square is a way to organize all these different possibilities. So you put the two gametes of one individual on top, you put the other two gametes of the other individual on the side, and then you fill in the boxes, right? All possible sperm genotypes on one side, all possible eggs on the other, and all possible zygote genotypes are placed within the square. So here, if a big T sperm manages to fertilize a big T egg, you would get a big T homozygous dominant tall plant. Suppose this big T sperm did not penetrate a big T egg, but it found an egg that had a little t. Well, it would still be tall, but it would be heterozygous. It would be a heterozygous tall plant. Suppose a little t sperm fertilized a big T egg. Right, then we would get another heterozygote that looks tall. And finally, if a little t sperm penetrated a little t egg, we would get a homozygous recessive short plant. So anytime you see a monohybrid cross, meaning you have two heterozygotes being mated together, we're going to get a genotype ratio of 1 to 2 to 1. Where for every one homozygous dominant, big T, there are two heterozygotes, big T, little t, and one homozygous recessive, little t. We always see a genotype ratio of 1 to 2 to 1 in a monohybrid cross, and we always see a phenotype ratio of 3 to 1, where 3 will be the dominant phenotype, tall, the dominant trait, and 1 will be the recessive phenotype, which would be short. And remember, if we counted 100 pea plants, that would mean 75% would be tall, 25% would be short. And this is by probability, right? It's possible to get 73 and 27. It's like flipping coins. It's possible if you flip a coin 10 times that you're going to still get 10 heads. It's possible, right? So these are just talking about probabilities, not what's happening. Just to review with another example from the book, suppose we do another monohybrid cross between two hybrid yellow seed plants. So we have two yellow pea plants that are both hybrid, meaning that they are heterozygous. They have one big Y, one little y, one big Y, and little y. And when we cross these, right, this female parent has two possible gametes. Right? It can either make a big Y egg or a little y egg. This male parent has two possible gametes. It can make a big Y sperm or a little y sperm. So now the Punnett square will tell us what are the different mixing and matching of sperm and egg. Okay, well, if this sperm fertilizes this egg, you'll get big Y, homozygous dominant yellow. If this sperm fertilizes this little y gamete, you'll have big Y, little y, heterozygous yellow. Over here, again, another heterozygous yellow, and then little y, you're going to have homozygous recessive green. This, again, shows us that 1 to 2 to 1 ratio. And again, that 3 to 1 phenotypic ratio. So just make sure those ratios make sense. And the ratios could be explained by the law of segregation partially. Punnett squares summarize meiosis and fertilization. Right, and could be explained by Mendel's law of segregation. So Mendel's law of segregation states that each individual has a pair of alleles for each trait, and that these alleles segregate, meaning they separate from each other during gamete formation, right? So when sperm and egg are made, we're no longer diploid. We don't have two copies of every allele. We have haploid sperm or egg. The sperm or egg will either have one or the other because the two pairs of alleles separated from each other, right? And again, we know that let's say we have a pure breeding yellow pea plant, that's big Y, big Y. When it grows into a plant and makes gametes, you're only going to have big Y sperm or big Y eggs, but they separated, they segregated from each other during gamete formation. And we know, of course, that's in meiosis. Right? In meiosis, we have the formation of haploid cells. Same idea. This green pea plant is pure breeding, so it could only give little y, little y. And each gamete, right, only has one allele from each pair. Each gamete only has one allele from each pair of alleles. It's only during fertilization, right, when two gametes, one from each parent, unite at random to become a zygote, and that's how we can get a hybrid plant. Right, so this is the law of segregation. It says that so even right now, this plant, big Y, little y, according to the law of segregation, this can make either big Y gametes or little y gametes, and the big Y and the little y can segregate. Right? So to make sure you're understanding this so far, let's do another example. This time, let's look at the flower color of these pea plants. So suppose you have a purple flower that are pure. This is a pure breeding purple plant, and this is a pure breeding white plant. And these two were crossed in the parental cross, the P generation. What do you expect the F1 to look like? Well, the F1 generation was 100% purple. Interesting. So these F1 flowers were allowed to self-pollinate to create the F2 generation. And what do you think the F2 generation looked like? Well, white flowers reappeared in some offspring, and they saw a 3-to-1 ratio, 3 purple flower to every 1 white flower. So 75% or 3/4 were purple and 1/4 were white. So now I want to draw Punnett squares to actually understand how we got here. Okay, so I'm going to use my little digital whiteboard. So let's come up with some abbreviations. Right? Let's say that big P is purple, where little p is white. I don't like to use P's usually because it's hard to keep track of lowercase and uppercase. But again, for now, let's just use this. So what would the genotype of this big P individual, or I just said it, of this purple individual be, right? It would be big P. So the parental generation, we have a big P purple flower. And what would this white flower be? What would the genotype of this white flower be? It would be little p. So this is our first cross. And so let's do this out. Let's do a Punnett square. So on top, we put big P. You could make a line if you want like that. So big P. And then we have little p. Right? So we can say, like, this flower over here can only give either a big P sperm, right? Or big P sperm. This little p over here can only give little p gamete or little p gamete. So now let's figure out what our possible offspring are. We have big P, big P, little p, big P, little p, big P, little p. What this says is that our F1 generation are all big P, little p. And what color should those be? Purple. So now let's just make sure this makes sense. So we have big P over here, little p over here. That means that these F1 individuals should be big P, little p, and purple. So that's actually exactly what we thought. Right? So now let's see what would happen if we take the first generation, the F1, and cross them with each other to get the F2. So now our F1 was big P, little p. And for our F2 cross, we have to self-fertilize these. So in this case, our Punnett square will have big P, little p. All right, that's one. And then big P, little p is for the other. So now let's put in all the different possible offspring. Right, so this big P can come with this big P over here. This big P can come with this little p over there. So hopefully you were able to do that in your notes. And now let's look, what are going to be the genotypes, the phenotypes to each of these. So big P, what color will that be? Purple. Big P, little p, what will that color be? Also purple, right? What about big P, little p? Right? That's going to be purple. And then what about this little p? Well, that is not going to be purple. So we have three different, three are going to be purple, and one will be white. So 75% purple, 25% white. And that makes sense given what we saw in this experiment. We saw that 3-to-1 ratio. So it's perfectly explainable by doing a Punnett square. So make sure you're able to do that on your own. So if you recall, I said that Mendel repeated these experiments not only with tall and short plants and round and yellow and green seed color, but with all these different traits. And in the F2 generation, he saw the same results from his monohybrid crosses. And look at all these numbers. It's quite amazing. He counted over 20,000 different plants, and combined all these traits gave the same ratio of 3-to-1 in the F2 generation. So this shows that the recessive trait that gets masked in the F1 generation reappears in the F2 generation, right? 1 to 3, right? 1 recessive to every 3 dominant. So it's important to understand what these letters, big P and little p, actually mean in biology. So we said the P gene, right? Or I didn't say it, yeah, but the P gene hypothetically controls flower color in pea plants. But there are 2 alleles of the P gene. There's a big P allele that encodes purple flowers and a little p allele that encodes white flowers. And we said that since purple is dominant to white, right, big P or big P little p will still give us the same purple flowered phenotype. But how does this make sense? Well, the P gene allele, right, the gene for flower color, makes a pigment making enzyme. So there's a gene for an enzyme that makes purple color. If you have an active version of that enzyme, we can call that big P. The big P version of this enzyme is active and produces the purple pigment. The recessive, the little p allele, is inactive. So this means there might be a mutation in that enzyme, so it can't make the purple color anymore. So it's inactive. No pigment is produced, and that's why it appears white. Right? Whenever there's a little p, that means it's a defective. It's a loss of function of that pigment-making enzyme. This little p cannot make purple color. This big P can make purple color. So let's look at this heterozygote. It still has a little p, but it appears purple because even one copy of the enzyme, big P, is enough to make the whole flower purple. Thus, big P, the purple allele, is dominant to the white flower allele. The recessive little p white flower allele is inactive. Maybe that's because of a substitution mutation or a frameshift mutation or something else. But regardless, it's a loss of function. That's why flowers with no pigment are white. Little p. In this plant type, we would say purple flowers are the wild type. since it's the most commonly found in nature. So the wild type allele is the most common form of a gene in a population. It's more than 1% by definition. So it's an important term. Wild type is the most common in nature. And if you're not wild type, that means you're a mutant. So the white flowers would be considered mutant, that are caused by a loss of function mutation, right? That enzyme can no longer make purple pigment. So there is a mutation in that enzyme, so it's not functional anymore, and that's why it's a mutant. So which of the following is a homozygous recessive genotype? I'm going to go through these very quickly. So right when I ask the question, you should pause because I'm going to give you the answer right away. The answer is C. In flying monkeys, gray fur color big G is dominant to white fur little g. Which of the following genotypes would lead to a gray phenotype? So pause here. And the answer is D. Right? Either A or C will give us a gray phenotype, which would give us a white phenotype. Right? Only little g would lead to a white phenotype. And of course, you have to be told what big G encodes for and which is dominant to which, right? You're not going to have to guess on an exam and remember which is dominant, which is recessive. In flying monkeys, right? If a purebred gray monkey were mated to a heterozygous gray monkey, what percentage of their offspring would be gray? So let's pause here, and I want you to draw a Punnett square right now that shows this mating. So hopefully you have the answer already in your notes, but let's do a Punnett square together. So a purebred gray monkey, let's stop there. When you have these kinds of problems, you want to break it down step by step. So let's write down what we know. A purebred gray monkey. What can we write down about that? Well, we know the genotype of that monkey must be big G, big G. Purebred means having the same two. They're homozygous. Gray means big G. And it's mated to a heterozygous gray monkey. What is a heterozygous gray monkey's genotype? Well, it has to have a big G because it's gray. And it's heterozygous, so it's big G, little g. So what percentage of their offspring would still be gray? So doing the Punnett square, this is what we get. And you can see that all of their offspring would still be gray. So the answer is 100%. Let's do another one. If a white monkey were mated to a heterozygous gray monkey, what percentage of their offspring would be gray? So now pause here and do the entire Punnett square and lock in your answer. So here's the Punnett square showing a heterozygous gray monkey mating with a white monkey. And here you see the answer. 50% would be white and 50% would be gray. So the answer is B. So up until this point, we've been seeing that a homozygous recessive genotype, right? So we saw, for example, little p gives us the recessive phenotype. So little p genotype gave us the white flower phenotype. But for individuals with a dominant phenotype, it's not as clear to figure out what the genotype is. So a purple flower can either be big P, homozygous dominant, or a purple flower could be big P, little p. So suppose I have a purple flower, and I want to know what the genotype is. Is it big P, or big P, little p? How do I figure that out? Well, I could use a test cross. A test cross determines the genotype of an individual having the dominant phenotype. So it says, is it a homozygous dominant or a heterozygous individual? The way we do this is by crossing the unknown dominant individual with a homozygous recessive individual, and then we look at the progeny. So here we have a purple flower. We have no idea if it's big P, or big P, little p, right? But we know it's the dominant phenotype. So what we have to do is a test cross, and a test cross takes this flower and mates it with a homozygous recessive flower. In this case, it would be a white flower. So I'm going to take this purple flower of unknown genotype and mate it with a white flower. And of course, a white flower has to be little p, little p. So now, if I mate this, and if it was big P, what would the results be? And if this purple flower were big P, little p, what would the results be? So this is called a test cross, because by crossing this unknown purple flower with a white flower and looking at the offspring, you can determine what genotype this flower was to begin with. Right? So if you get all purple offspring, that means it must have been big P, right? And that's how you get 100% purple offspring. If this were heterozygous, though, you would get 50% white, 50% purple. So by doing a test cross, you can look at the progeny and figure out what the parent's genotype was. So pause here and answer this question now. And the answer is A. Right? Must have been big P, big P. Let's say you observe 50% purple offspring. What was the genotype of the purple parent? The answer is B, big P, little p. And again, you should do these Punnett squares in your notes because you'll have to do them on the quiz and exam. Here's a sample problem I'd like you to try right now. So in cactus color, you're told green, big G is dominant, yellow is recessive. You have a yellow father and a true breeding green mother. So pause here and answer these 3 questions, and I'll show you the answer. So the genotypes of the parents, right? We have a yellow father, that must be little g, and a true breeding green mother must be big G, right? What colors are the children? Well, if you have that kind of a mating, all of them will be big G, and they'll all be green. And then in that mating, you'll see that 25% will be yellow, little g. And then this question, you should see that there's a smooth father, bumpy mother, and the children will be 50% smooth, 50% bumpy. If these don't make sense, try to go over it yourself first, but then flag it and bring it to recitation when we go over these problems. Right? Recitation is critical for this upcoming quiz because you have to ask your specific questions so you know how to do these genetics problems. So let's take a break from plants and talk about human genetics. And phenylketonuria is a human genetic disorder. Phenylalanine is one of the 20 amino acids naturally found, and phenylalanine is found in many foods. So most of us break it down using an enzyme called phenylalanine hydroxylase, and we have no problem at all. However, some people have a mutation in this enzyme. So phenylalanine cannot be broken down. And if there's too much phenylalanine, there can be nervous system disorders. And that's where phenylketonuria results from. So how many copies, assuming that you're fully healthy, how many copies of phenylalanine hydroxylase do you have in each of your cells? The answer is 2, right? You have 2 copies, one from mom and one from dad. A phenylketonuric is somebody who lacks any functional enzyme. Okay, it does not have any functional phenylalanine hydroxylase enzyme. So this means phenylketonuria is a recessively inherited disease. That means somebody has to have 2 copies of the recessive mutant gene in order to have the disorder. For example, even having 1 copy of this enzyme, phenylalanine hydroxylase, say we'll call that big P, that's enough to break down phenylalanine and be healthy. So whether you are big P, or big P, little p, you can still break down phenylalanine and be just fine. Only if you're little p, homozygous recessive, do you have not 1 functional copy of this enzyme, so phenylalanine accumulates. So we would call the little p version the mutant allele. The big P version would be the wild type allele, since that's more common. Okay? So even having 1 copy of the normal gene is enough to be healthy in a heterozygote. Right? And again, the normal gene is referred to as the wild type since it's found in greater than 1% of the population. The non-functional copy of the enzyme would be a mutant, little p. And when we're born, our heels, now it's a little more sophisticated, but for many, many years, a little heel prick would be done on a newborn, and then the blood would be sequenced or analyzed to see if it can break down phenylalanine. And if there is a mutation in the phenylalanine hydroxylase gene, then that baby would not be able to be fed any food with phenylalanine, right? Because then it would cause nervous system disorders. So the parents have to make sure the baby who is a phenylketonuric does not consume any phenylalanine. And Diet Coke has a label. Phenylketonurics contains phenylalanine, right? Aspartame, which is the sweetener, contains phenylalanine. So if you're a phenylketonuric, meaning you do not have this enzyme, you cannot drink Diet Coke. And it's not good for you anyway, so you shouldn't be drinking too much, regardless. So Punnett squares are useful to track the inheritance of genetic disorders like this, and also like cystic fibrosis. So just like PKU, phenylketonuria, cystic fibrosis requires you to have two bad copies, two mutant copies of the CFTR gene. The CFTR gene normally allows chloride out of the cell. Even having one copy of that is fine you're healthy enough. But if you have two mutant copies of CFTR, that means you have cystic fibrosis. So we call that a recessively inherited disease. We can call the healthy CFTR gene big F and the mutant CFTR gene little f. So somebody who is big F is completely healthy. Somebody who's little f has cystic fibrosis. Right? Somebody who's healthy has two copies of the good CFTR protein. Somebody with cystic fibrosis has not one good copy of the CFTR protein. However, if you're a heterozygote, big F, little f, we call them healthy carriers. They're healthy because even one copy of the CFTR gene is enough to not give them cystic fibrosis, but they still carry that mutant cystic fibrosis allele. They're a carrier. So this comes into play when if we look at two carriers, that means there's a chance that they have a child that will have cystic fibrosis. So in diseases, we call the heterozygotes carriers because they carry the diseased allele, but they're healthy. So let's try to do this type of Punnett square. If two healthy carriers for CF have a child, what is the chance the child will be affected by cystic fibrosis? So affected means will have it. And two healthy carriers means they're big F, little f. So pause here and draw a Punnett square. So if the mother is a healthy carrier, that means half of her eggs would have big F, half of her eggs would have little f. If a father is a healthy carrier, half of his sperm will have big F, half of his sperm will have little f. We know that what is the chance of a random sperm, one of these sperm, meeting one of these random eggs? Well, this sperm can meet this egg and give us big F, big F. This sperm can meet this egg and give us another healthy carrier. This sperm can meet this egg, another healthy carrier. Or this unhealthy sperm can meet this unhealthy egg, and then we get cystic fibrosis. So the answer is A, 1/4 chance that the child will be affected by cystic fibrosis. And I just want to note that probabilities are not actualities, right? So we're going to say 25% chance that the child will have cystic fibrosis. And that's just pure probability, right? It's 50% chance of the man's sperm having little f, 50% chance of the female's egg having little f. So just probability that it's 25%. It could be higher, right? It could actually be two children. The first two children both have cystic fibrosis, and that would defy probability. So Punnett squares can advise us, like in which direction probability is likely to take us, but it doesn't tell us exactly what's going to happen, right? Punnett squares don't predict the future. So up until now, we've been looking at one trait at a time, but it's also possible to track the inheritance of two traits. So Mendel wondered how different traits are inherited together. So he tracked each of two traits through different generations. So he started with the parental generation that were both true breeding, but they differed in two separate traits. So now we're not just looking at seed color or seed shape, we're looking at seed color and seed shape. So he took a female plant that was green and wrinkled. So it's little r, little y, little y. And then he took a yellow and round male pea plant, right? Big R, big Y, big Y. Again, these are both true breeding, right? Because big R, big Y, big Y. They're both the same allele. So these are both two true breeding parents, and they differ in two different traits. This one is green and wrinkled. This one is yellow and round. So now we have to think about what kind of sperm and egg can this female give? So what kind of egg can this female produce? Well, we know that every gamete has one copy of each gene. So it's going to have a little r and a little y. It's the only possibility, right, that this egg can have is little r and little y. Looking at this male P, the only gametes that could be made from here are big R and big Y. Therefore, the only possible offsprings would be big R, big Y, little y. So the F1 plants show both dominant characteristics because they have both big R and big Y. And therefore, they're going to all be yellow and round. Right? If we were to self-pollinate the F1 plants, then we would get the F2 generation. So now we're going big R, big Y, crossed with a big R, big Y, little y. And that is what we call a dihybrid cross. Right? A dihybrid cross makes hybrid organisms that differ in two different traits. So this is showing a dihybrid cross. Big R, big Y, big R, big Y, little y. And in a dihybrid cross, two genes on different chromosomes can be combined into one large Punnett square. So instead of 4 boxes, we have 4 by 4. So 16 different boxes. So the important thing to know from this type of individual is that you could get 4 different gametes. Right? From this individual over here, you could have 4 different gametes. A way to remember that is using FOIL. I don't know if you remember that from algebra. First, outer, inner, last. So basically the gametes, any possible R could match with any possible Y in this female. But every female egg is going to only have one R and one Y, right? Because these are gametes. One R and one Y. So the gametes can either be first, so it could be big R and big Y. First. To the first letter. It could be the outer letters, so it could be big R and little y, that's over here. It could be the 2 inner letters, little r, big Y, and that's shown here. Or it could be the last, little r, little y. And of course, the male has the same exact genotype, so the sperm will have the same 4 combinations. And all you're doing in a dihybrid cross is putting these two together. So if a big R, big Y sperm meets a big R, big Y egg, you're going to get an offspring that's big R, big Y, big Y. And again, you just fill in these boxes. And it shows the same ratio every time you do a dihybrid cross. You get a phenotypic ratio of 9 to 3 to 3 to 1. This is something you must remember. A 9 to 3 to 3 to 1 ratio is always seen in the F2 generation of a dihybrid cross. What that means is 9 out of 16 will have both dominant traits. 9 out of the 16 will be round, which is dominant, and yellow. So you can count those to be 9 boxes that are round and yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's the 9 part. Then we have 3 that are going to be dominant in one trait, but not the other. So there'll be 3 that will be yellow and wrinkled. Yellow and wrinkled, yellow and wrinkled, yellow and wrinkled. And there are going to be 3 that are round and green. So that's the 3 and the 3. And then finally, 1 out of 16 will be both recessive. Will be both wrinkled and green. So this 9 to 3 to 3 to 1 ratio is found anytime you have a dihybrid cross. It'll be a little trickier, and I'm not going to give you a complicated dihybrid cross to solve. But you have to understand this ratio, how to understand what gametes this individual can make, and again, understanding what a dihybrid cross actually is looking for. So based on the results from his dihybrid crosses, Mendel proposed a second law of inheritance. And the law of independent assortment states that the segregation of alleles for one gene does not influence the segregation of alleles for another gene. In other words, right, the alleles for a single gene do not influence the alleles of a second gene. They are independently assorted into gametes. So suppose we have this dihybrid individual that's yellow and round, but it's big R, big Y, little y. That means, right, this is a duplicated cell. We have big R, little r, big Y, and a little y. And of course, there's sister chromatids to each of these because these just went through interphase. In meiosis, we know that after crossing over occurs, there's random alignment of homologous chromosomes on the equator of the cell. But we don't know how they're going to orient themselves. It can be that this big R and big Y are on top and little r and little y are on the bottom. And if that were to happen, then in meiosis 1, we get this. And then we get gametes that are only big R, big Y, or little r, little y. But we also know that at metaphase, it's possible for them to mix and align differently. So it's possible that there's big R and little y, and then little r and big Y. And that's just random, right? Random alignment on the metaphase plate. And of course, in that situation, we're going to get gametes that are combinations. We're going to have big R, little y, right? Or little r and big Y. So this is what allows all possible combinations of alleles in gametes. Right. So this is again how let me come up with an example. So your blood type, right, has nothing to do with your skin color, right? When your skin color genes separate, your blood type genes don't influence your skin color genes, right? They do that independently. They assort independently from each other. As you'll see in a little bit, the law of independent assortment does not apply to genes that are very close together on the same chromosome. So there's an exception. So when two genes are very closely together on the same chromosome, they do not independently assort because they're too close. Actually, where one gene goes, the other will likely follow because they're so close. That's called linked genes, and we'll talk about that later. Now's a good time to watch the video on dihybrid crosses. Shown in the ebook. And again, talking about those ratios. So let's do an easy dihybrid practice problem together. In cats, the allele for gray fur, big G, is dominant over the allele for beige fur, little g. The allele for solid coat, big S, is dominant over the allele for a striped coat, little s. If you have a pure breeding solid and beige cat and you cross it with a pure breeding striped and gray cat, that's our question. First, what are the parental genotypes? So pause here and tell me what the parental genotypes are. Next, I want you to state the genotypes and phenotypes of the F1 cats that are produced as a result of this cross. And here are the answers. So here we have a solid big S is solid. Little g means beige versus a striped gray cat. So here's our genotypes and phenotypes. This cat over here let's assume this is the female cat. This can only make one type of egg, right? This egg has to be little g, big S. This male cat over here can only make one type of sperm. Big G and little s. Therefore, when you put these together, the F1 will always be big G, big S, little s. And since solid is dominant over striped and gray is dominant over beige, all the F1 cats will be solid and gray. Now, I want you to calculate the phenotypes resulting from a cross of a pure breeding solid beige cat and one of the F1 offspring. So pause here and try to figure that out now. Right, so in this case, we're mating little g, big S, with a dihybrid big G, big S, little s. So to make the Punnett square, we have to see, okay, what kind of gametes can this person make? What kind of gametes can this person make? Well, this one we already know can only make one type of gamete, little g and big S. Right, that makes it easier for us. But this one over here, this guy, big G, big S, little s, the F1 can make 4 different types of gametes. And remember foil. It could be the first letters, big G and big S, right? And again, this is because of mixing and matching, just like we saw independent assortment, right? This big G can go with the big S, or this big G can go with the little s. So that's what big S. I'll draw my little s's like that. Right, so we can have big G, big S, big G, little g, big S, or little g, little s. Those are the 4 possible gametes that this F1 individual can make. So now we just have to do the Punnett square. This is an easier one. So if this sperm, or this egg, went with this sperm, what would you get? What would you get here and here? You're going to get 4 possibilities, 4 possible genotypes. So let's see what we get. The answer is we would get these 4 possible genotypes. Anything that has big S, since everything has at least one big S, that means they're all going to be solid. But these little g's will be solid in beige, and these will be solid in gray. So the answer is 50% solid in gray, 50% solid in beige. This is the hardest type of problem you might be asked on an exam. So we could also use laws of probability to help us a little bit when we have some of these kinds of genetics problems. So let's review some probability basics before continuing on. If you flip a coin, what is the chance that you land on heads? Right, so hopefully you said C, 50%, or 1/2. If you roll 1 die, what is the chance that you would roll an even number? So pause here. And since a die has 6 sides, 2, 4, and 6, 3 of the 6 are even. If you roll 1 die, you have a 3 out of 6 chance of getting an even number, which equals 1/2, or 50%. So the answer is D. So now that we understand the basics, we can introduce what's called the product rule. The product rule tells us what is the probability of 2 independent events occurring together. So the product rule says that the probability of 2 independent events occurring together is the product of their individual probabilities. So you multiply the probability of one thing happening times the probability of a second unrelated thing happening, and that will give you the likelihood that both things will happen. The product rule is also known as the AND rule because it tells you what is the likelihood of one thing happening and another thing happening at the same time. So what is the chance of rolling a 4 on a die 2 times in a row? This is another way of saying what is the chance of rolling a 4 and rolling another 4? So if the chance of rolling a 4 once, the first time, is 1 out of 6, right? And if you roll the die again, the probability of getting a 4 is still 1 out of 6. So then you multiply the probabilities of each. So it's 1/6 times 1/6 equals 1 over 36. So you have a 1 in 36 chance of rolling a 4 2 times in a row. And that we just figured that out by looking at the individual probabilities, right, and multiplying them. And they're independent, right? If I roll a 4 once, it doesn't make it less likely for me to roll a 4 the next time. It's just as likely every time I roll the die for me to get a 4. So it's 1/6 times 1/6. So pause here and answer this question now. And the answer is, again, A. If you roll the die twice, what is the chance that you would roll an even number the first and the second time? So what is the chance that you're going to roll it? So pause here. I really want you to try to think this out on your own. Don't just get lazy and just let me tell you all the answers. If you roll a die once, the chance of getting an even number is a half. If you roll it again, the chance of the even number is a half. So what is the chance of rolling a die twice and getting an even number both the first and second time? Right? It would be 1/4. 50% times 50% is 25%. Right? It's always important to remember how to add and subtract, multiply, and divide fractions. So 1/2 times 1/2, right? 50% times 50% is 1/4. You multiply all the numerators on top and all the denominators on the bottom. Another way to think about it's 50% means it's half. So what's half of 50%? 25%. So let's apply the product rule in a genetics problem. So suppose we have a cross between two heterozygous individuals. What are the odds of getting a little a individual in the next generation? So you could draw a Punnett square, of course, big A, little a, big A, little a. And we know that if they're both heterozygous, there's a 50% chance of getting a little a from the father and a 50% chance getting a little a from the mother. So there should be a 50% times 50%, or 1/2 times 1/2 equals 1/4 chance of getting both a little a from mom and a little a from dad. And by doing the Punnett square, we get the same answer, right? 1/4. So the product rule just reflects probability, reflects what we're going to see in the Punnett square. Again, from across a big Y, little y, big Y, little y, what is the chance of getting big Y offspring? Well, the chance of each box, right, represents 1/4 because you're multiplying 1/2 times 1/2. There's a 50% chance of a big Y sperm, 50% chance of a big Y egg. 50% times 50% equals 25% chance of getting a big Y. Right? So that's, again, is explaining the product rule. 50% chance, 1/2 to get a little y, 1/2 to get a big Y. So what is the chance of getting a big Y and then a little y? Right? It's 1/2, or sorry, I should say it's 1/4 here and 1/4 here. And that gives us a total of 50% would be heterozygous. So really quickly, I want to just tell you, you do not have to know how to do this complicated type of problem. But from a dihybrid cross, big R, big Y, little y, big R, big Y, little y, what would the offspring be? So you can actually, so you remember the 9:3 to 3:1 ratio. You can automatically just predict 9 will be yellow, will be both dominant. 3 will be one dominant, not the other. 3 will be one dominant, not the other. And then 1/16 will be both green and wrinkled. Another way to do this kind of instead of doing a big Punnett square, you can break it down into two separate monohybrid crosses. So we can look at big R, little r. And then the Y's separately. And we'll know, right, just phenotypically, once we do this, I'll do this quickly right now. So if you want to figure out how many of these will be yellow and round, instead of doing the whole dihybrid cross out, you can break them down into two separate Punnett squares and say, okay, so what is the chance of it being round and yellow? So what is the chance of it being round? 1, 2, 3. 3 out of 4. 3 out of 4 will be round. How many will be yellow? 1, 2, 3. 3 out of 4 will be yellow. So what is the probability of getting a P that is round and yellow? Well, for that we use the product rule. So we multiply the individual probabilities of a round P and a yellow P. And by multiplication, we get 3 times 3 is 9. 4 times 4 is 16. And 9/16 is our magic number that we said, right? 9/16 is what we always see in a dihybrid cross of being both dominant traits. And again, you could say, what is the likelihood of getting a wrinkled and green? So this is 1/4 times 1/4, which gives us 1/16. So again, I'm presenting things in a lot of different ways, hopefully not confusing you, but once you find the right way that you like to solve genetics problems, you'll be fine. Right? There's multiple ways to solve a genetics problem. I'm also going to continually point you back to the end of the section in the book where it has a whole guide on how to solve genetics problems in the back of chapter 10. So you must review those very carefully. The other rule you have to know is the sum rule. And the sum rule is the probability of either of two mutually exclusive events occurring. So what is the chance of this happening or that happening? To do that, we have to take the probability of either of two events and add them together. So what is the probability that event 1 or event 2 will occur? Right? So this is saying, what is the probability of getting a 3 or a 4 when you roll a die? So for this, we're using addition. The sum rule is addition. The sum rule is also known as the OR rule because it's when you're getting a 3 or a 4. So what is the probability of getting a 3 when you roll a die? Well, it's 1/6. What is the chance of getting a 4? Also 1/6. What is the chance of getting a 3 or a 4? When you hear or, that means plus. That means the sum rule. So the chance of getting a 3 or a 4 is 2/6 or 1/3. So we now reviewed the product rule and the sum rule. The product rule really comes in handy when solving some more complex genetics problems. So suppose we have a question: if two parents are heterozygous for 3 genes, R, Y, and T, what is the probability that their child will also have the same exact genotype? This seems confusing, right? It's like, oh, where do we start? But it's easy if we just figure out, we make 3 separate Punnett squares for each of these genes, and then we just multiply the likelihood of getting each of the combinations. So I want you to try to do this on your own, so please pause here. Right, and hopefully you were able to use the product rule. And we can make 3 separate Punnett squares. So let's just do the monohybrid cross with R, big R, little r, big R, little r. And we're asking, what is the likelihood of getting an offspring that has this genotype? So what is the likelihood of getting big R, little r? Well, 50% chance. What about the big Y, little y? Well, again, 50% chance. What about big T, little t? Oh, again, 50% chance. So what is the likelihood of getting big R, little r, and big Y, little y, and big T, little t? Well, it's 1/2 times 1/2 times 1/2, or 1/8. So that's how the product rule makes it a lot more simple. So pause here and try to answer this question now. And again, it's the same logic. So you just do 3 separate Punnett squares. So you could just take the example of 1, big Q, little q, little q, little q. And you're being asked, what proportion will be homozygous recessive for all 3 genes? So here you're going to see 50% will be homozygous recessive for Q. If you repeat the same thing with the B's, you're going to say 50% is homozygous recessive for B. If you do the same thing for the D's, you're going to say 50% are homozygous recessive for D. Therefore, what proportion of the offspring will be homozygous recessive for Q and B and D? The answer is again 1/8 D. So Punnett squares and the product rule assume that 2 genes are sorting independently. That means they're on separate chromosomes or very far away on the same chromosome. When genes are linked, that means that they're very close together on the same chromosome. They're physically near each other. And when genes are linked, we cannot use the product rule and Punnett squares and expect them to reflect reality. Right? They do not assort independently. Linked genes are not inherited independently of each other. They're inherited dependently of each other. Right? When one gene is inherited, a linked gene is likely to be inherited as well. So it's not like independent assortment. It's not random probability. Thomas Hunt Morgan at Columbia in the 1900s studied a pair of traits in Drosophila, which is fruit flies. He looked at body color and wings. And he took a homozygous recessive fly with a heterozygote. And normally you would expect a 1:1:1:1 ratio of all these different combinations if the genes assorted independently. But what he saw was that most of them were either heterozygotes or homozygous recessive. So the two genes, the gene for body color and wings, were likely inherited together. Right? So when you had a gray body, you were more likely to have normal wings. When you had a black body, you were more likely to have vestigial wings. That means that the two genes must be very, very close together and therefore are inherited together. So Punnett squares in the product rule cannot be used if genes are linked, because inheriting one allele will influence the likelihood of inheriting a linked allele. This is why a lot of people with red hair have freckles. Like, it's a simplified example, but the red hair gene is close to the freckle gene, so to speak. So therefore, it is very common that if you do have red hair, you also have freckles. And genes can become unlinked during crossing over. We know that genes on the same chromosome do not necessarily travel together because during prophase 1, crossing over between homologous chromosomes mixes and matches alleles. So it's possible that during meiosis, we get a chromatid that'll have both big A and big B, just like one of the parents did, or we can have what we call a recombinant chromatid, which has one from mom and then the other from dad. So these two are recombinant because they look like neither parent originally. These two are parental because they resemble what the parents originally looked like little a, little b, little a, little b, big A, big B, big A, big B. The recombinants are found in neither parent. And of course, the recombinants can only be formed because of crossing over. And when two genes are very close together, the likelihood of crossing over is very slim. Right? What is the chances of getting a crossover right here in the perfect spot? Not very likely. The probability of a crossover event between two linked genes is proportional to the distance between the genes. Basically, the farther apart two genes are, the more likely crossing over is to separate them. These two genes, W and Y, are so close together. Therefore, when W is inherited, Y is also likely inherited together as two linked genes. But since V is far away from Y, it's possible that crossing over here can give you a big V and then little w, little y, and then little v and then big W and big Y. And it's possible by looking at these crossover frequencies to map out the distance of genes on a chromosome. And that's what you'll talk about a lot more in a genetics class. So to finish with part 1, I'd like you to look at the textbook. Page 214 and 215 has a whole guide on how to solve genetics problems. So go through that and then start practicing some problems in the back of the book before moving on to the next bit. So thank you for listening, and I hope you learned a lot about genetics before we move on.